The challenge was, could there be polymorphisms involved that influenced that outcome? Could there be a reason that the early part of the trial looked like NAC was effective, but then disappeared? And given those two genes were located very close to each other, we selected 5 polymorphisms that were located in TOLLIP that we knew were functional. And that's what this slide shows you. It shows you polymorphisms, DNA locations that influenced either the promoter region, the exon, which is the actual product level, or the microRNA binding site, which also limits production. And we knew that these SNPs were involved in susceptibility and survival and function. So we took a look at those and we added in the MUC5 promoter SNP because of its proximity and of its high influence and outcomes. And we conducted genotype counts and frequencies using a chi-squared or Fisher's exact test to look at the drug-gene interaction in a multivariable Cox regression model, applying a bond for rodent correction. To get enough events, we used a composite endpoint of the death, hospitalization, and transplant or decline in FVC. And then we used an additive model for the TOLLIP polymorphisms, and a dominant model was used for all other SNPs. Now, in the Panther data set, 341 patients were enrolled, 315 were eligible for the analysis, 154 enrolled in the genetic substudy, giving us 54 patients assigned to the placebo arm and 60 assigned to the NAC arm. While there were no differences in the pre and post, I am showing you on the demographics what this population looked like, and they were even across the board. Really no differences involved in any of the traits between any of the arms. And at the bottom of the slide, we see the genetics of those polymorphisms laid out in those arms with no real differences. And so we know that in the non-genotype group, again, no differences were observed. So if we had powered this for the entire group, we would have had a much greater power to determine even more polymorphisms. But as we looked at the drug-gene interaction table, we still successfully hit several polymorphisms. Most importantly was RS3750920 in TOLLIP that had a clear interaction with N-acetylcysteine. There were also suggestions just missing the mark in 2 others, one in TOLLIP and one in the MUC5 polymorphism with a p-value of 0.06 and 0.07 respectively. With 2 other genes hitting the mark or just missing the mark, we went on to a replication cohort to confirm our findings. And what we found was that we were able to confirm them in the original 3750920. Now what's most fascinating is if you take a look at the panel on the left, you see that NAC was actually harmful with a hazard ratio of 3.23, whereas in the panel on the right, it was helpful with a hazard ratio of 0.14. Translating that ratio, you were 7-fold less likely to die or hit the composite endpoint with the proper polymorphism, and 3 times more likely to die with the other polymorphism. Now, what does this translate into? The CC polymorphism represents the homozygote major. That's 25% of the populace. The CT is the heterozygotes at 50%. And the TT is the homozygote minor at 25%. So in other words, a quarter of the patients benefited greatly from NAC, whereas a quarter of them were harmed, giving us a flat representation that we saw in the study results. When we took a look at the replication cohort, as we pointed out, only the 3750920 held up with very similar hazard ratios of 5 and 0.22. So what were our conclusions or takeaways? The NAC may be effective in 1 out of 4 patients. And this would be the first pharmacogenetic interaction ever demonstrated in IPF. No real clinical traits were noted to be different between the group that was genotyped and not genotyped. And there were certainly no genotype differences by treatment arm or enrollment period. And that we were able to replicate these findings really becomes supportive of trying to look at a future trial. These findings really supported an ongoing universal genotyping in future NIH trials, such as cleanup.